Hi students, welcome to KR Microbiology classes. So today the topic is we are going to start with Immunology part 3. We are going to learn about Louis Pasteur, scientist, and two another topics, attenuation and virulence. Okay, so you already know very well about the Louis Pasteur. He is a French chemist. He is very well known scientist in the microbiology, and he also discovered the fermentation process, and he coined the term vaccine. And you know that vaccine means it came from the Latin word vacca, right? Vacca means cow. And he also worked on so many kinds of diseases like pebrine, anthrax, chicken cholera, rabies. He worked on these diseases, and he also discovered attenuated vaccine for the rabies, and he also discovered attenuated vaccine for the chicken cholera. Okay, so I'm telling again and again one word. Have you found it? మీరు పట్టుకున్నారా నేను ఏం వర్డ్ని again and again చెప్తూ ఉన్నాను? That is attenuated vaccine. So here you have to get that doubt, what is attenuation, right? So attenuation means the process of removing virulence of a pathogen without affecting its antigenic properties. It is known as the attenuation. So తెలుగులో చెప్పాలి అని అంటే ఏదైనా సరే ఒక పాథోజన్ యొక్క డిసీజ్ కాజింగ్ కెపాసిటీని తీసేసి యాంటీజెనిక్ ప్రాపర్టీస్ని తొలగించకుండా కేవలం అది డిసీజ్ని కలుగజేసేటువంటి వ్యాధి తీవ్రత కలుగజేసే శక్తిని కనుక మనం తీసేస్తే దాన్ని అటెన్యుయేషన్ అని అంటాం. ఓకేనా? The process of removing virulence, so that pathogen definitely have the capacity to elicit or to stimulate the antibody production in the host, but it don't cause any disease. Means its disease causing capacity will be decreased or removed using certain processes. That is known as the attenuation. Here you will get one doubt, madam, what is virulence, what is pathogenicity, and what is antigenicity? So see here, pathogenicity you already know very well. In pathology you already came to know this word, or in microbiology or immunology also very well known word, it is pathogenicity, right? Pathogenicity means the capacity to cause disease is known as the pathogenicity. Virulence, సో పాథోజెన్సిటీ అంటే వ్యాధిని కలిగించే శక్తిని మనము పాథోజెన్సిటీ అంటాం. విరులెన్స్ అంటే how severely, virulence means how severely the disease will be caused. That capacity is known as the virulence. ఎంత ఎక్కువగా డిసీజ్ని కలుగజేయగలదో ఆ కెపాసిటీని మనం virulence అంటాం. And what about the antigenicity? The capacity to stimulate the antibodies in the host, that capacity is known as the antigenicity. Means ఎంత ఎక్కువగా హోస్ట్ బాడీలో యాంటీబాడీస్ ప్రొడక్షన్ని స్టిమ్యులేట్ చేయగలదో ఆ కెపాసిటీని మనం యాంటీజెన్సిటీ అని అంటాం. ఓకే, అండ్ నౌ we have to understand first how Louis Pasteur came to know about this attenuation process. So here this is very interesting thing, that as a scientist he is working on the vaccination process. So he is doing many experiments at that time. He introduced this Pasteurella aviseptica into the chicken, right? So but after some time he observed whether the disease was produced in this chicken or not. But surprisingly this chicken didn't got any disease or illness or any symptoms of that particular disease. He was surprised why the disease was not caused in the particular chicken. I was introduced that bacteria, no, so నేను ఆ బ్యాక్టీరియాని ఎక్కించాను ప్యాథోజన్ని, బట్ వై ఎందుకు దానికి వ్యాధి రాలేదు అని ఆయన సర్చ్ చేశారనమాట. సో హీ స్టడీడ్ మోర్ అండ్ మోర్ అండ్ హీ చెక్డ్ ఎవ్రీథింగ్ వాట్ హీ డిడ్. Then he came to know that he introduced that bacteria belongs to the old culture. So he came to know that okay, so for in the time being, so కాలం గడవడం వల్ల దాని యొక్క వ్యాధికారకమైనటువంటి శక్తిని అది కోల్పోయింది అని ఆయన తెలుసుకోవడం జరిగింది. సో ఇన్ ద టైమ్ బీయింగ్ that Pasteurella aviseptica, that particular pathogen lost its disease causing capacity. So he came to know that. So by this experiment he first understood this process attenuation. Later he did few more experiments. So how we have to do the attenuation? So by changing the temperature also we can remove one pathogen's virulence. మనం ఒక pathogen virulence ని టైం బీయింగ్ తోటి మాత్రమే కాకుండా టెంపరేచర్ తోటి కూడా మనము అటెన్యుయేట్ చేయొచ్చు. ఎలా చేయొచ్చు అని అంటే, see here he also did another experiment, that is what he did, he cultured two cultures of Bacillus anthracis bacteria. He taken two culture plates and he cultured Bacillus anthracis same strain in the two culture plates. One was incubated at 42 degrees centigrade and another one is at the 37 degrees centigrade. One was at the 42 degrees centigrade and another one is the 32 degrees centigrade. Sorry, 42 degrees centigrade and 37 degrees centigrade. In this 42 degrees centigrade plate, and these bacteria grown and these are introduced into the sheep. These are introduced into the sheep. Surprisingly, the bacteria which were grown at 42 degrees centigrade, they didn't cause any disease. And the bacteria which were incubated at the 37 degrees centigrade, they caused the disease. So by this, he came to understand that, so by increasing the temperature also, we can attenuate the pathogenic bacteria or pathogens. And he also did another experiment. This time he used the introduction of these pathogens into the unnatural hosts. So you have to know that what is this unnatural host. Okay, so if I will tell you in Telugu, temperature విషయంలో ఏం చేశారంటే నాన్న, బ్యాసిలస్ యాంథ్రాసిస్ బ్యాక్టీరియాని తీసుకొని ఆ బ్యాక్టీరియాని రెండు కల్చర్ ప్లేట్స్లోకి తీసుకున్నారు. ఒక కల్చర్ ప్లేట్ని 42 డిగ్రీస్ సెంటిగ్రేడ్ దగ్గర ఇంక్యుబేట్ చేశారు, ఇంకొక కల్చర్ ప్లేట్ని 37 డిగ్రీస్ సెంటిగ్రేడ్ దగ్గర ఇంక్యుబేట్ చేశారు. సో ఈ 42 డిగ్రీస్ దగ్గర ఇంక్యుబేట్ చేసినటువంటి కల్చర్ ప్లేట్ని ఒక షీప్లోకి ఎక్కించారు. దాంట్లో ఎలాంటి డిసీజ్ రాలేదు. బట్ 37 డిగ్రీస్ సెంటిగ్రేడ్ దగ్గర ఇంక్యుబేట్ చేసి గ్రో చేసినటువంటి బ్యాక్టీరియా ఏదైతే ఉందో అది కూడా ఇంకొక హెల్దీ షీప్లోకి ఎక్కిస్తే దానిలో డిసీజ్ అనేది కనిపించింది. దీని ద్వారా అర్థమైనది ఏంటి అని అంటే, టెంపరేచర్ ఇంక్రీజ్ చేయడం వల్ల ఒక బ్యాక్టీరియాకి డిసీజ్ కాజింగ్ కెపాసిటీ అనేది పోయింది. ఐ మీన్ విరులెన్స్ అనేది పోయింది, అటెన్యుయేషన్ అనేది జరిగింది అనేది అర్థమైంది. సో ఇన్ ద నెక్స్ట్ వన్ ఈజ్ ద introducing the pathogen into the unnatural host. The name, it has the meaning unnatural host. So for every pathogen there will be some natural host, right? So for the smallpox virus, if you see human beings, they are the natural host. So what he did, he introduced the smallpox virus into the cow and he introduced the rabies virus into the rabbit. So by doing these things, those virus, they lost their virulence capacity, the disease causing capacity. So by these processes, he came to understand attenuation process and he introduced this attenuation vaccine for the rabies also using these processes. And nowadays another process is also there, using genetic engineering. By manipulating the genes also we can attenuate the pathogens. So this is all about the today topic, Louis Pasteur, his findings, his achievements, and attenuation process, virulence and pathogenicity. I hope you understood this topic. If you have any doubts, please mention in the comment section. Thank you very much. Thank you for watching.